Thank you. It's really an honor to get to speak with you today about our research, and I just want to say how grateful I am for the warm welcome into your scientific community. I think I can speak for all of ASM when I say that. My work deals with herpes viruses, something we haven't heard so much about yet today. These viruses are the masters of persistence because they're really the masters of our cell biology. They're very complex DNA viruses who have evolved with us, co-evolved with us, so they know our biology better than we do, and they're wonderful teachers of cell biology. So there are 9 herpes viruses that infect humans, and I'm going to tell you about our work on the beta herpes viruses, which has been changed here, but cytomegalovirus. And so all herpes viruses establish this lifelong latent infection. And most of us carry these viruses asymptomatically in a healthy host, but there are possibly some risks to being seropositive for the virus as well as possibly some benefits. But the real danger with cytomegalovirus comes from an immunocompromised or immunodeficient state, and this is really prevalent in the context of a congenital infection. CMV is the leading cause of congenital disease and really outnumbers the much better known and understood congenital defects or syndromes, and this affects really a large number of babies worldwide. CMV is also a serious problem in the context of its reactivation in the immunosuppressed or immunocompromised, which was really important in the HIV, early days of HIV and its outbreak, but also today still a very high cause of morbidity and mortality in transplant patients, either solid organ or stem cell. Now, in order to get a handle on CMV disease and controlling it, especially in the transplant setting, we really have to understand its latency and how to control its reactivation. So latency for herpes viruses across the herpes virus families have been defined as the maintenance of viral genomes, restricted viral gene expression, and the absence of progeny virus production. But in the case of CMV, we actually really know very little about the nature of the latent infection. We can't identify an exact profile of transcripts that are important for the latent infection or some restricted small number of transcripts that are important for latency. And there's microRNAs that are important, but this is really very much still a black box. So what the field's been focusing on is understanding host restrictions and chromatinization of the genome for silencing for latency, host signaling and the transcriptional milieu present in the cell that gets infected that is going to impact latency, and modulation of host processes that are important to latency. And what I think a picture that is beginning to emerge is that following some differentiation or stress, which can occur with in reactivation of any herpes virus, there's a change in cellular state or factors that is going to have to climb to a threshold to cross a checkpoint to result in changes in viral gene expression, again having to meet a threshold, and a fairly high threshold, to then ensue a commitment to viral DNA synthesis and an active reactivation to really tip that scale. So my lab has been really focused on understanding how the virus is sensing and responding to these changes in the cell. This requires carefully filtering out the noise to make good decisions to either maintain your latent infection or to reactivate it. And so to do that, we have started by trying to identify viral genes that might be important to this sort of decision making, and then the host processes that they are interplaying with. And so this is just a schematic of the viral genome. You have a unique long region and a unique short region flanked by repeat sequences. And we began to focus on a unique region of the genome called ULB prime. This was only discovered in 1996 when they sequenced the first clinical strains of the virus out of humans. It's 15 kilobases of sequence and encodes nearly 20 open reading frames that had never been known to exist. And so for decades, we had studied CMV in the absence of this region. The first gene we discovered to be important was UL138, but when we sequenced the RNAs, it's encoded on long RNAs that encode 3 other open reading frames, and none of these genes had ever been studied. Latent infection, following the commitment to DNA synthesis, we get additional transcripts that really focus on the expression of UL136 and UL138. So we made the decision that we needed to study all these genes together because their expression was coordinated and they're probably acting together. Each of these genes encodes multiple proteins, both membrane-associated and soluble forms. They're highly disordered. AlphaFold can do nothing with any of these genes. They lack homology to any known proteins in any of the databases, and so that, of course, there are no orthologs to any of the other herpes virus genes. And they're dispensable for replication in fibroblasts, but they do have functions in other cell types. So I'm just going to tell you a little bit about the genetics that we have spent a lot of time trying to understand and understanding the function of these genes. The UL138 gene is a pro-latency gene in that it suppresses virus replication. So that means when we mutate it, the virus actually replicates better. UL135, very surprisingly, was pro-reactivation or pro-replication. In fact, we couldn't get a virus that lacked UL135 to replicate at all unless we disrupted also UL138. So there's an antagonism between these two genes. So we knew this genetic antagonism had to be based on some function of the proteins, and indeed that is in the regulation of the epidermal growth factor receptor, where UL138 is promoting or potentiating the signaling of EGFR. This is pro-latency, and UL135, by recruiting host E3 ubiquitin ligases, can attenuate EGFR signaling, and this is very important for reactivation. But what I want to talk to you about today is UL136. UL136 is an incredibly complex gene. It makes proteins that are very difficult to work with, but they seem to have this really interesting ability to toggle back and forth between a 138 or a 135 dominant state. So 136 was really launched in the lab by a courageous graduate student, Katie Cavanis, but this is a story I'm going to tell you that has occurred over a decade has really been carried forward by some very talented and wonderful scientists. So the first thing we do when we want to study one of these genes is we engineer on a C-terminal MIC tag. That's because initially we didn't have antibodies. We do have antibodies to the proteins at this point, but not for 136 because it really has no immunogenicity. We've tried to make antibodies 3 or 4 times. Anyway, when Katie did the first infections with a recombinant virus expressing the MIC tag on UL136, we very surprisingly got this ladder of proteins. We weren't sure what to do with those. So the next thing that we do, the second virus we always make, is engineering in stop codons for any 5' ATGs so that we abrogate translation of the protein. And in that case, we lost the expression of this top isoform, but we maintained the expression of everything going forward. It took a lot of recombinant viruses, but eventually Katie determined that each of the protein isoforms here is derived from initiation at a different methionine codon along the coding sequence. So this gene is actually expressed as these sort of natural N-terminal truncations of the UL136 gene. She then put these viruses into fibroblasts, and as you can see, there's really no phenotype. They all replicate very similarly to the parental virus that just encodes UL136 expressing the MIC tag. The only exception is this little increase in virus replication in a virus that lacks the 23 and 19 kilodalton isoforms of 136. These are soluble isoforms that we haven't yet separated genetically. So what about in latency? So I want to tell you a little bit about the systems that we use to understand latency. We use human primary hematopoietic progenitor cells that are marked by CD34. We infect those cells with a virus that we have engineered to express GFP as a marker for infection, and of course those recombinant viruses can also house any gene disruption or alteration that we want. We infect these cells, we isolate out the infected cells using GFP, and we put them into a long-term bone marrow culture over a stromal cell support, and then culture them for a period of time to allow the virus to establish latency. We're then going to compare the infectious centers that are produced prior to reactivation in the time that the virus should have established latency versus following a reactivation. And this is what those phenotypes look like. So a wild-type virus establishes a latent infection, very few to no infectious centers, but we can induce reactivation and then get infectious centers out. You'll see loss of latency phenotypes. This is the kind of phenotype that a UL138 mutant virus has, in which this virus is just replicative. It can establish a quiescent infection, and you make just as much virus prior to a reactivation stimulus as you get from reactivation. We also have viruses that fail to reactivate, and this is the case of a UL135 mutant virus. And then in the case of UL136, these viruses all span the entire gamut of phenotypes that we can expect from this, and I'll show you that data in just a minute. But another model system that is really important is one that we developed with the late Jane Nelson, who was a fantastic mentor and friend, and Patrizia Capozio at Oregon Health and Science University. And this is using humanized mice, where we infect the mouse, we humanize the mouse with human CD34 hematopoietic progenitor cells that we've derived from bone marrow or cord blood. We then infect fibroblasts and transfuse these fibroblasts into the mice. They will seed the infection of the human CD34 positive cells. We need to use humanized mice because the human virus is so restricted in its tropism. There is no animal model for this virus, and this is really the best we can get. We allow the virus to establish latency, and then we stimulate mobilization of those cells, differentiation of those cells, and hopefully reactivation of the virus. And we get very similar phenotypes to what I just showed you in vitro. Viruses that fail to reactivate, viruses that just replicate and fail to establish a latent infection, and some viruses that even replicate better than the parental viruses, like these are viruses you cannot shut off. So here's what the data looks like for the UL136 mutants. The UL136 parental virus establishes latency. A virus that lacks the largest isoforms, the 33 kilodalton or 26 kilodalton isoform, these fail to reactivate. And viruses that lack the 25 or the 23 or 19 kilodalton isoforms all have more replicative phenotypes. And then something we commonly see is in a null mutant virus that doesn't have these isoforms that can promote replication or promote latency, you just get a virus that doesn't seem to know what it's going to do. It can't really establish a quiescent infection, but it's also not very responsive. In the humanized mice, we get very similar phenotypes. We have viruses that fail to reactivate. They don't respond at all to stimulus, and we don't detect an increase in genomes and tissues. Or viruses like the 2319 that is a more replicative virus and replicates even without a reactivation stimulus. The difference between these assays come with the 25 kilodalton mutant virus or the null virus, in that these viruses really aren't responsive to the stimulus in mice, whereas they were more replicative in the in vitro infections. And so these are differences in the context of infection that we don't yet understand, but are really interesting. So in doing this work, we were really noticing how difficult these proteins were, and half the time we ran blots, they looked like this, where we were missing some of the protein bands for UL136. And so we did a cyclohexamide treatment to just see if some of these bands were unstable, and sure enough, the 33 kilodalton isoform is very unstable. We can rescue that by proteasomal inhibition and regain expression of the the levels of 33 kilodalton protein. And over a cyclohexamide chase, looking at the decay of the protein, you see that the half-life of this isoform is very, very short, only an hour, relative to the stability of the other isoforms, which is really rather long. So there are 4 lysine residues. We know the protein's being targeted to the proteasome, so we assume it's a ubiquitin-mediated pathway. So there are 4 lysine residues in the entire coding sequence, and 3 of them are unique to the UL136P33 isoform. We mutated those to arginine, thinking that we could stabilize this form, thinking that the instability of this protein was probably important in viral infection. And so sure enough, by lysine to arginine mutation, we can get a protein that is stable, and we put that into fibroblasts, and it replicates just like the wild-type virus. It doesn't matter at all. But go back to the context of latency, and that virus now has this loss of latency phenotype where it replicates the same with or without a reactivation stimulus, but it is kind of a wimpy virus in that we really don't get a whole lot when we stimulate it. It really is a little bit stuck. And you'll kind of notice on some of these graphs, there are really large error bars. When we produce any virus that has lost the ability to establish a latent infection, it's just replicative, the error bars get big because there's a lot of noise that is entered into that system, we think. But that error collapses right down when we create these viruses that can respond, which we find really interesting and something we don't fully understand. We get the same phenotype in the humanized mice, and that the K to R mutation has this loss of latency phenotype, and the virus can only replicate. So we wanted to know what cellular factor was targeting P33 for turnover, because that tells us what cellular pathways the virus has evolved sensitivity to in order to regulate its decisions between latency and reactivation. And so We commonly use IP mass spec and yeast-2 hybrid both in combination to identify cellular factors. And in this case, we identified IDLE, which is the inducible degrader of LDLR. It's an E3 ubiquitin ligase that can target LDLR, VLDLR, and ApoE-R2 for degradation. And so this protein is a very important protein that is it's a therapeutic target for controlling hypercholesterolemia. And it's induced transcriptionally by the liver X receptor, which is induced by oxysterols. So the question is, can it target P33 for degradation? And so this is just an IP validating the interaction between IDLE and P33. And then we wanted to know, can IDLE actually degrade P33? So if we overexpress IDLE with P33, you see the loss of P33. However, oops, this is all messed up, but this is the K to R mutation. IDLE has no ability to target P33 for degradation when the lysines have been mutated to arginine, and this is just quantitated here. So what about if we induce the expression of IDLE with either an LXR agonist or an inducer of IDLE? Do we see degradation of P33? And so this is infection in fibroblasts, and you do see this, lowering of levels of P33. But if P33 has been stabilized with the K to R mutation, you don't see any effect, and that's quantitated here. One thing I'll just point out that we don't yet understand, when we stabilize P33, it really changes the expression of these middle isoforms. So there's also regulation going on of 136 by this protein. A third model that we often use to understand latency is the THP1 monocyte cell line model. And this is a good model, but it's far from perfect. It models the restriction of viral gene expression for latency and the re-expression of viral genes following reactivation. We can get very synchronous reactivation, which we cannot do in the primary human bone marrow cells. It's amenable to CRISPR, which the primary cells really are not. But it doesn't support a full replication of the viral genomes or the production of infectious progeny. But this model has been really important for us to come to understand how virus-host interactions are pivoting around differentiation because we can get that synchronous reactivation. And so we looked at IDLE expression in these cells, and you see that IDLE by these white bars is very highly expressed in the undifferentiated cells. In infection, we see a reduction in IDLE transcripts that isn't statistically significant unless you're looking at the K to R mutant virus where P33 has been stabilized. So this suggests that P33 is actually feeding back to affect the expression of IDLE in ways that we don't yet understand. When we differentiate these cells into macrophages using a 4-ball ester, you see that IDLE levels just fall to the floor. It doesn't matter about infection anymore. So this allows us to hypothesize that the instability of P33 is important to the establishment of latency, where IDLE is very highly expressed in undifferentiated cells, but differentiation is going to lower the levels of IDLE and allow for P33 levels to rise and accumulate. Now, as I said, we don't have an antibody for P33, and in hematopoietic cells, P33 isn't even expressed high enough levels that we can detect with an epitope tag. So I'm going to use the immediate early proteins, NUL135, as expression of which would be a proxy for a virus that is going to replicate and is far more active. And so I'm showing you here in the yellow where we have now knocked down IDLE with CRISPR, and this should then allow P33 levels to rise, and this should drive virus infection forward. And so you see much higher expression of IE1 and IE2, where we have knocked down IDLE. At 1, 3, and 5 days, it begins to go down after that. And then in the stabilized form of P33, in that infection, you see just equivalent levels of IE1 and IE2, very high levels, and the knockdown of IDLE makes no difference. So we've stabilized P33, so it doesn't matter if you have IDLE or not, and that's just quantitated here. This is the control for the CRISPR. So in the primary hematopoietic cells, we used the drug, the LXR agonist, GW3965, to induce IDLE to basically enforce this instability of P33. When we use this drug, you can see IDLE transcripts go up, and we now have a virus that doesn't reactivate from latency. So that really suppresses the reactivation from latency if you can't get this accumulation of P33. And of course, in the virus that has the stabilized form of P33, there's no effect of the drug on reactivation in that case. So what I've shown you is that in the context of latency in undifferentiated cells, LXR signaling is high, idle levels are high, and this keeps a primary determinant for virus reactivation low. Upon differentiation or some stress, idle levels will fall, which allows P33 levels to come up. P33 is going to feed back and it drives viral gene expression. We've also shown that once the virus commits to virus replication of the viral genome, that really drives the higher expression of the transcripts that give you more P33, and this may even perhaps feed back to reduce idle further. And so this basically tells us that the virus has evolved to be able to a sensitivity to cholesterol signaling in ways that we really want to understand how that's important and how that impacts viral infection in the host. And this fits into a much bigger picture in which we're beginning to understand many virus-host interactions that are important to latency, UL138 driving EGFR, driving transcription factors that feed back and reinforce expression of UL138, 135 that's overcoming all of that, and now the regulation of 136 needed for reactivation, keeping it low in the context of latency, higher in the case of reactivation, which then can result in other virus-host interactions that drive immediate early gene expression and replication. And so I would just like to thank Tsinghua University and China for a really warm embrace of us having us here. This has been a wonderful trip, and acknowledge the people in my laboratory who really contributed so mightily to this work. Thank you.
Thanks. Any questions?
Nice talk. So you showed 136 and 138 is important to regulate reactivation or latency, respectively. So what is the underlying mechanism? For example, the 138 is in charge of latency, right? And is function linked to the gene silencing?
Yeah, that's a great question. So, you know, the herpes virus field has been very focused on epigenetic regulation of the genome, but there's a lot of signaling that happens upstream of that. And so we know for latency the genome has to be silenced. So some of the next steps would be to understand how some of the signaling, whether it's EGFR signaling or other downstream signaling pathways, the regulation of cholesterol homeostasis, how that could actually be impacting chromatinization of the genome, and we're just not quite there yet.
Okay, any questions still? Minghua. Any microphone?
Yeah, this is really hard. So we could do it in humanized mice, perhaps, and we just haven't done that yet. The humanized mouse model is pretty hard enough. But you know, MCMV doesn't have any of these genes. As I said, there's no orthologs in any of the other herpes viruses. Chimpanzee CMV has these genes, but rhesus doesn't even have these genes, and the murine virus doesn't. So we're really kind of stuck there in terms of animal models. It's been one of the hard aspects of this.
MCMV doesn't have this day hallmark.
No, MCMV does not.
Okay, sorry for that.
Okay, thanks. Thanks, Felicia. Thanks for a wonderful talk.